Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive. Today we're looking at something really quite groundbreaking in cancer therapy. Basically, researchers have found a new way, using AI, to arm T cells against cancer. And the key takeaway, the really big point, they've built this rapid, designed-for system that creates custom proteins to target cancer mutations inside the cell, stuff we previously thought was, well, completely out of reach for therapies like antibodies. And they can do it in weeks.
Exactly. That speed is critical because, current therapies like CAR T cells, they're great, but mainly for targets sitting on the outside of a cancer cell. The problem is many of the most important cancer-driving mutations are inside the cell.
Right, they're intracellular proteins.
Yeah.
But they do get advertised, don't they, through these little flags on the surface?
That's right. Tiny fragments of those internal proteins get presented by complexes called pMHC class I, peptide-MHC complexes. They're like little signals saying, look what's inside.
But these signals, these pMHCs, they're notoriously difficult targets. Flat, small, antibodies just don't bind them well, is that it?
Precisely. The binding surface is just too tricky for traditional, larger biologics, like antibodies, to get a good, specific grip. You risk hitting healthy cells if the fit isn't perfect. So this paper's big idea was, let's not try to adapt old tools. Let's design completely new, small proteins, they call them minibinders or miBds from scratch, computationally to fit perfectly.
Quick custom design keys for these very specific locks. So how do they actually build these keys using AI? Let's walk through Figure 1, the design pipeline.
Well, it's a pretty slick computational workflow. They start with generative AI models like RFdiffusion, which kind of dream up thousands of possible protein shapes, the basic scaffold.
Just the shapes initially.
Yeah, the blueprints. Then another tool, protein-MPNN, figures out the best amino acid sequences to actually build those shapes. And AlphaFold 2 predicts how stable the final structure would likely be.
That must generate a huge list of possibilities.
Yeah.
How do you filter that down from thousands to something you can actually, test in the lab?
That's where the smart filtering comes in. Two key steps. First, something they call in silico cross-panning. It's like a digital check to make sure the potential binder won't accidentally stick to similar-looking healthy peptides.
Okay, so weeding out the potential off-target effects early. What's the second filter?
Molecular dynamic simulations, MD. Think of it like putting the virtual protein binder through its paces, testing how it moves and flexes, especially right at the point where it binds the target. They looked for binders with the least amount of wobble, the lowest energy fluctuations when bound. That's how they pinpointed the best candidate, NY1B04. It significantly reduced the chances of picking a dud for lab testing.
That makes sense, testing its physical integrity virtually before making it real. Okay, so they picked NY1B04. Figure 2 confirms it binds really tightly in the lab, right? About 6.9 nanomolar affinity.
Very high affinity. And cryo-EM imaging actually confirmed the structure looked almost exactly like the computer prediction. The design was spot on.
Okay, so here's the question then. We've got this computer-designed molecule, NY1B04. It sticks like glue to the target flag. But can you actually weaponize it? Can it make a T cell kill a cancer cell?
That's the crucial next step, the functional proof in Figure 3. And the answer was yes, absolutely. They engineered this miB, NY1B04, into a standard SAR construct, basically bolting it onto the T cell machinery. They called these IMPACT-T cells.
IMPACT-T cells, okay.
And when they put these IMPACT-T cells together with melanoma cells that displayed the target NY-ESO-1 peptide,
It worked.
They worked beautifully. The IMPACT-T cells effectively found and killed the target cancer cells, showing the miBds successfully redirected the T cells.
Wow, so the computer design part really did function as a targeting system for the T cell. And you mentioned that cross-panning check earlier. Did that hold up in real tests too?
It did remarkably well. The in silico predictions about which similar peptides it wouldn't bind to were correct for 8 out of 10 variants they tested experimentally. So the digital filtering was reliable.
Okay, that's impressive validation. Now, Figure 4 seems to push this even further into personalized medicine, right? Targeting neoantigens.
Exactly. This is maybe the most exciting part. Neoantigens are those unique mutations specific to an individual patient's tumor. Often, like the RVTDESILSY target they used, there's no experimentally determined structure available.
So how do you design a binder if you don't even have a precise map of the target?
You predict it. They used AlphaFold 2 first to predict the 3D structure of the neoantigen peptide bound to its MHC molecule. Then they fed that predicted structure into their miBD design pipeline.
And it still worked, even starting from just a prediction.
Yes, the pipeline successfully generated and identified 2 promising miBds against that neoantigen target. It shows the whole approach is potentially scalable even for brand new patient-specific targets where you only have sequence data to start.
This feels like a real shift then. Moving immunology from screening lots of candidates hoping for a hit to actually designing the solution you need up front.
It really is. The innovation is this repeatable, fast, design-first platform. 4 to 6 weeks from target identification to a validated binder candidate that's incredibly fast. It essentially cracks open that whole class of intracellular targets presented by pMHCs.
Yeah, so the implications are, well, huge, right? Beyond just NY-ESO-1.
Absolutely. The immediate thought is personalized cancer therapy, targeting those unique neoantigens in each patient. But you can imagine using this for infectious diseases too, designing binders against specific viral peptides presented on infected cells, or even for autoimmune diseases, maybe designing binders to block the T cells that mistakenly attack healthy tissues. It gives us a powerful new way to engineer specific biological interactions.
Okay, let's try and boil this down. 3 key takeaways for you listening. First, AI-driven computational design can now create these highly specific minibinders, or MHCs, to effectively target those tricky pMHC flags on cancer cells targets that were largely inaccessible before.
Second, integrating tools like molecular dynamic simulations for, let's call it digital stress testing, and structure prediction tools like AlphaFold significantly speeds things up and makes the process much more likely to succeed, finding the best candidates faster.
And third, this whole platform works even for personalized targets, neoantigens, where you might only have a predicted structure to start with. That really opens the door for tailoring therapies.
It's a very powerful platform, but of course there's always a what's next or a potential challenge. These miBds are totally novel proteins designed de novo. As they move towards the clinic, a key question will be immunogenicity. Could the patient's own immune system see these therapeutic miBds as foreign and attack them? That's a safety hurdle that will need careful navigation.
A fascinating point to consider. Designing the therapy is one thing, making sure the body accepts it is another. That brings us to the end of this deep dive. Join us next time.